Hi, my name is Alister DaCosta. I'll be talking to you today about some of the work that we've been doing on detecting chromosomal translocations using augmented genome sequence graphs. I'll start off by talking about some current approaches to structural variant detection, including translocations, and some of the limitations that we've found to these approaches. I'll then be talking about pangenome graphs, including the direct augmentation of sequence graphs, before presenting SOMVARG, somatic variants from graphs, and showing how we can use our tool SOMVARG to detect somatic translocations in tumor samples. Starting off with the way that structural variants are now commonly detected. We most commonly detect structural variants, especially those in long reads, based off of the alignment of reads to a reference genome, say GRCH38. Once we've got read to reference alignments, we can interpret these alignments to identify if the pattern of alignment supports a type of structural variant, whether that be an insertion, a deletion, an inversion, a tandem duplication, or some form of larger scale chromosome rearrangement. One of those larger chromosomal rearrangements is interchromosomal translocations, where a portion of one chromosome is exchanged with that of another, shown here at translocation between chromosomes 4 and 11. We can see that at the breakpoint fusion, where the two chromosomes have actually translocated and swapped sequence, we can see that there's a possible gene fusion, where one gene from one chromosome has merged with that on the other end with the other chromosome and generates a fusion gene product that may be detrimental to the health of the individual where this translocation has taken place. The way that we identify translocations from long reads is very similar to that of other structural variants. We look for read to reference alignments that are split, specifically looking for a portion of a read to have a portion of it aligned to one chromosome and another portion of that read aligned to a different chromosome. And if there are a number of reads that have the same pattern of split alignments to the same regions of the genome, we can infer a possible translocation between these two genomic regions based off of the alignments of the reads to those regions. Overall, reference-based structural variant detection works quite well, but it is limited, especially with samples that contain non-reference mobile element insertions, such as the line 1 element shown here. A sample contains a 6,000 base pair line 1 element, and we got a 10,000 base pair read taken from the sample. When we align this read to the reference GRCH38, we expect it to align end-to-end, and as the line 1 element present in the sample is not present in the reference, we expect to see an insertion in this alignment. But what we often see is the observed alignment at the bottom here, where a portion of the read aligns as expected, the flanking sequence, not the line 1 mobile element sequence, but the genomic flank that matches the reference. But the genomic insertion sequence, the line 1 insert sequence, does not align as an insertion. It aligns somewhere else to some existing line 1 copy in the genome. And with the prevalence of mobile elements in the genome, lines and allos and other repetitive sequences, there's a lot of copies for it to align to. And what happens if we get a number of reads that have the same pattern of split alignment here is we could get a false positive translocation. So we look to quantify how often that this pattern of split alignment could arise, because a consistent alignment pattern between two regions may lead to false positive translocation calls. We used reads from the HPRC, the Human Pangenome Reference Consortium, specifically from sample HG00438. They generated some nanopore R9.4 reads, and we aligned these to GRCH38 as well as to the diploid assembly of the sample HG00438 that they had generated using HiFi ASM and PacBio HiFi reads. When we aligned these reads to GRCH38, we got about 203,000 reads that had possible split alignments. That is, they had multiple high-quality alignments to different regions of the chromosomes. And what we saw that when we aligned the same reads to the diploid assembly was that we only got 126,000 reads that had split alignments, as many of the reads that did contain mobile elements present in the diploid assembly but not present in the reference were able to align end-to-end through that area of known variation. So the key point that we're trying to make is that, and what we're seeing is that when we align reads to a sequence that contains known variation, if the read also contains that variation, it's going to align better compared to aligning to a sequence that doesn't have that. And diploid assemblies are great, but they're not always available. There are limitations to being able to generate a diploid assembly, whether that be cost or sequencing coverage or depth or technology. And one of the things the HPRC has shown is that there's a lot of shared variation between individuals. So the HBRC has recently generated and published a draft human pangenome reference, and they've taken a number of diploid assemblies, generated a number of diploid assemblies from a vast array of individuals from different genomic backgrounds and across populations, and used tools like minigraph cactus, and pangenome graph builder to build pangenome graphs that contain paths for known variations seen in these individuals, but not seen in our base linear reference sequence. And one of the things that they highlighted was that a majority of the variation that they see present in these individuals that's not in the reference is actually shared by at least 2 individuals. And these pangenome graphs that they generate are sequence graphs. Each node contains a sequence, and we can approximate each of the contigs used in the graph by taking a path through the graph based off of and sort of sticking the nodes together. So you can see the blue path here, the green path, and the yellow path. The nodes, if you take the nodes in the sequence based off of take the sequence of the nodes together, you can approximate the haplotype that's used here. And pangenome graphs are a great representation of the diversity across the human population to look at compared to just a standard single linear reference. Now, there's a lot of known variation that's contained in these pangenome graphs, right? We've got 47 different individuals' data contained in these graphs, and there's a lot of variants that are unique to 1 or 2 individuals that are here that might be shared by others in the population. So we can align reads to these graphs now using tools like minigraph and graph aligner. And that alignment allows us to not only consider the base GRCH38 sequence at the time of alignment, but also other sequences, other areas of known variants, other known variant sequences that are present in the population, but that are not present in GRCH38, but that may be present in some of these reads, allowing us to reduce the number of split alignments and increase the number of end-to-end alignments. So we can look at these 4 reads and see that we've got 4 end-to-end alignments, some of them through nodes that contain known variations, some of them maybe through just the base reference. And overall, we can say that a pangenome graph allows us as a better alternative to a linear reference for translocation detection, because we get reduced numbers of split alignments, especially ones that would lead to false positives. But if we do have a read that genuinely contains sequence from 2 different chromosomes, we would still expect it to align with a split alignment. That is, it would align to a portion of it would align to nodes that represent 1 chromosome, while a portion of it would align to nodes that represent another. And we can still interpret this pattern of split alignment from reads to a pangenome graph alignment and infer translocations from it. And this is the core of what our tool SOMVARG is doing, somatic variants from graphs. It's a tool that is looking to detect interchromosomal translocations based off the alignments of reads to a pangenome graph rather than reads to a linear reference graph. As mentioned, the pangenome graph, or a sequence graph, augmented sequence graph, allows us to contain known variants within the graph that are not present in GRCH38. SOMVARG finds split alignments across different chromosomal graph segments, non-overlapping on the read, both with a mapping quality 60 and a combined alignment length of at least 50% of the read. And if we get 3 reads that have the same pattern of split alignments, we can consider that a candidate translocation. So the HBRC has shown how pangenome graphs can be generated from a set of diploid assembly contigs, and existing pangenome graphs do represent a diverse range of individuals and a lot of known variation, but each individual themselves is going to have some variants that may not be present in the pangenome graph. And when it comes to cancer detection, we often have paired tumor normal samples, a normal sample from that individual and a tumor from that individual. And it's not always feasible for us to be able to generate a diploid assembly from that normal sample. But we've come up with a method to be able to try to leverage some of the information that's present in that normal sample and use that to augment a graph without having to go through the full diploid assembly process. We can use existing reference-based approaches to identify variation in a normal sample and take those known variants and apply them directly to the reference to generate an augmented sequence graph. And we can then take this graph and align tumor reads to it, and in the same way that we would align reads to a pangenome graph, because this is in effect the same graph structure and format. It's a sequence graph. And we can look at the paths that those alignments take. And as this graph contains variants that are known to that individual's normal, any variants that are unique to the tumor would show up here as, say, split alignments for translocations, but any variants that are shared between the tumor and the normal the reads would just align through those paths for known variants. So we applied this approach to a set of cancers, specifically mixed lineage leukemias, as part of a collaboration with Dr. Tracy Murphy and Dr. Mark Minden at the Princess Margaret Hospital. Translocations in mixed lineage leukemias typically occur at a known 8.3 kilobase region on chromosome 11, with one break end occurring in the MLL/KMT2A gene. And there's a large number of possible other ends for these translocations. About 80 other distinct genes have been observed as translocation partners with the KMT2A MLL gene for translocations in MLLs. So identifying the precise breakpoints of MLL translocations can be used for residual disease monitoring. And our lab has been working with Princess Margaret Hospital to get precise breakpoints that can be used to design PCR primers, hence our motivation for actually identifying translocations. I'll be using these MLL samples to be able to compare SOMVARG's detection ability to that of current reference-based approaches. So the data that I'm going to be using to do this is a subset of a larger cohort. This subset includes 16 MLL samples, each with one cytogenetically verified translocation, 10 normal samples from cultured normal blood cells, two of these normal samples are matched to some of the these 16 MLL tumors, while 8 others are normals from another other samples in the cohort of the study. Each of these samples were sequenced with an ONT promethyne with an R9 flow cell, and we can see here a short overview of the normal samples, and similarly with the tumor samples, noting known translocations between chromosomes 9 and 11, 10 and 11, 4 and 11, as well as 11 and 19 present. So these are the cytogenetically verified translocations that we're looking to detect. We then took the 10 normal samples, we sequenced them and identified insertions present using existing reference-based approaches, pooled these identified insertions, and used our approach that I described earlier to directly augment a graph with these known variants. So this augmented graph was we term it the 10 sample augmented graph. We then took, for each MLL tumor, its nanopore reads, and we aligned them to both our 10 sample augmented graph, the HPRC's pangenome, and to GRCH38, using either minigraph or minimap2. We then used SOMVARG or Sniffles2 to call translocation calls, whether it be graph-based or linear reference-based, and we then compared them together. So looking at the number of calls made, we expect there to be one call made for sure, as that's the expected cytogenetically verified translocation call in each of these samples, but we don't necessarily know for sure if there's going to be more. We don't expect there to be a large number of translocation calls in these samples, maybe just a couple, maybe a handful. We don't expect hundreds, tens to hundreds of translocation calls. What we can see here is the number of calls made by Sniffles is an order of magnitude higher than that made by SOMVARG, and whether it be with the 10 sample graph or the HPRC pangenome graph. So we can see, regardless of the coverage level, in all of the samples that we looked at, we were able to identify the expected translocation with all 3 methods, but the number of calls made with SOMVARG is drastically lower. This is a log scale, so there's an order of magnitude reduction in the number of calls made by SOMVARG compared to Sniffles2, where we believe the majority of these calls made by Sniffles2 that are not made by SOMVARG are false positives. Not just the majority, we think all of them are. When we look at the amount of time that it takes to detect translocations running each of these tools on a single thread, we can see it again, SOMVARG is much faster than Sniffles2 as well. So it's able to reduce the number of false positive calls, what we believe to be false positive calls, and do so in a faster time. We then looked at another sample that's been well studied, COLO829. It's a melanoma. Previous study with the New York Genome Center that's been published identified 18 somatic translocations in the sample using Illumina short reads. So we sequenced both the COLO829 tumor and the COLO829 normal with Oxford Nanopore R10.4, 33x normal, 61x tumor. And we then took the normal samples, identified insertions in them, and generated an augmented graph from this single normal set of insertions that almost acts as a pseudo-personalized reference for this sample. We then took the tumor samples reads and again aligned them to this augmented graph, to the HBRC graph, and to the linear reference using minigraph and minimap2, respectively, calling translocations with SOMVARG and SNFLs2. We use the New York Genome Center's Illumina-based call set as our truth set here, and we compared translocation detection with SNFLs2, SOMVARG against the HBRC graph, and SOMVARG against our matched normal graph. We can see that the recall was slightly higher for SNFLs2 here compared to SOMVARG and the match normal, but the number of false positives, and thus the precision was a lot higher with SOMVARG compared to SNFLs2, an order of magnitude reduction in the number of false positives. Some of our future work here includes expanding graph augmentation beyond insertions, looking at deletions, inversions, and duplications, also bringing in multi-threading support. We also are hoping to expand variant detection beyond just translocations to look at somatic indels and other structural variants from read to graph alignments. In summary, just to conclude here, I know I've talked a lot here, so we'll wrap it up. We present methods to directly augment sequence graphs from a set of known variants when a diploid assembly is not available or feasible. We also present SOMVARG, a tool for the identification of interchromosomal translocations from augmented sequence graphs, and we show how SOMVARG improves precision and decreases detection time. And we note that SOMVARG is available on GitHub here, and it is available for your use. With that, I'd like to thank members of Simpson Lab, the U Lab, our collaborators at Princess Margaret Hospital, and thank you so much for listening and taking the time to take a look at my presentation. Hope you have a great day.